मैंने ही डाली थी वो तो काफी स्टोरी रिपीट
Good evening, Ishika. How are you doing? Okay. So, in the last class, we had conversed about the fact that how transcription occurs, and I told you about the acquainted, made you acquainted with the sigma factor also. Today we'll be discussing more about that transcription, okay, in detail. How does it happen, and how are the promoter sequences recognized by the specific RNA polymerase in eukaryotes? They are recognized by the transcription factors. Okay. So before going into that detail, I'd first tell you something regarding the nomenclature of the DNA and pertaining to your mRNA. Okay, so DNA is a double-stranded molecule. We are all aware of it. So when we denature the DNA, okay, just I'm not denaturing it. I'm just linearizing it so that the double helix can make you perplexed. So what I'm doing is, suppose this is your DNA. Okay. Now let's assign this 5 to 3 prime anti-parallel, 3 to 5 prime. This is our DNA molecule. Now let's assign an arbitrary sequence. Now let's make it a complementary sequence. Now the mRNA that is being produced, okay, when the mRNA is being produced, it uses this strand as the template strand. Now you will ask why. Because the thing is, all the polymerases in this entire world, all the enzymes that are known as polymerases, they are the ligase enzymes. They make the phosphodiester bond or the catalyze the formation of the phosphodiester bond. They are never catalyzing from 3 to 5 prime direction. They're always catalyzing from the 5 to 3 prime direction. So when you will be using this as a template and the two strands are anti-parallel, then you will be able to make the anti-parallel of that, which is 5 to 3 prime. So the mRNA being produced will be a 5 to 3 prime mRNA, which would have used this as the template. So there are reasons pertaining to when I'll teach you DNA repair the exonuclease activity. In that chapter, I'll explain also to you why 3 to 5 prime polymerization would be ineffective, would be you can say lethal mistake committed by evolution. That's why that has not been committed, and 5 to 3 prime polymerase, you can say polymerase activity has been inculcated in the polymerases. One more thing, You must have wondered sometimes that why do I write like this 5 to 3 prime, 3 to 5 prime? Why are they not parallel? Okay, why both the strands are anti-parallel? The reason is when you go for parallel, suppose both the strands are 5 to 3 prime. This is a valid question. Suppose let's make this. Why is DNA not like this? Why both the strands are anti-parallel to each other? Why not both the strands stay parallel to each other? So this alignment is not present in nature. Why? The answer to that question also is there. Okay, since biology you have answers to every question that you ask. Nothing is to be mucked up. So the answer to this question is collinear hydrogen bonding. Okay, when we see or when we observe the hydrogen bonding between A and T, G and C, that hydrogen bonding is completely collinear. Means I'll just show you the image. Collinear will provide more stability rather than haphazard zigzag hydrogen bonding. So let's see DNA anti-parallel strands. If you see images, you can just see the image of collinear hydrogen bonding. So you can just click on any of these images, you can see this is 5 to 3 prime, this is 3 to 5 prime. Okay, you can see that bonding is collinear, straightforward bonding, and this would not be a collinear bonding. Okay, if you see parallel strand, DNA parallel strand hydrogen bonding, I don't know if the image will be present or not. Mm, so this is regarding the anti-parallel. I think this may be the image. You can see this image, this one. You can see if we have, okay, if we try to make this strand 5 to 5 prime, what you will see is you will see some kind of bend. Okay, not real. This will not be a collinear hydrogen bonding. Then what you will see is, okay, you will see some instability in the hydrogen bonding. Okay, so that you will see there. In case of anti-parallel, the hydrogen bonding will be very, very collinear. So like in protein biochemistry, when I will teaching you, so I will be teaching you the beta strand. So again, anti-parallel beta strands are more stable because again of collinear hydrogen bonding. Okay, that is why DNA molecule, the rubber strand molecule, are anti-parallel in nature. Both the strands are anti-parallel. They are bounded by hydrogen bond anti-parallelly. Now, when 5 to 3 prime mRNA is produced, this is being used as a template. So what will be its sequence? A, U, G, C, G, C, C, U, A, G, G, C, U, A. Now, what is the difference between this sequence and this sequence? Don't they look identical? They do, except for this one. T is replaced by U, means thymine is replaced by uracil since it's an RNA. So only T is replaced by uracil. Otherwise, the entire sequence is identical. Okay, keeping this in mind. Okay, keeping this in mind, historically, scientists have given or bestowed some terminologies upon both these DNA strands. This is known as minus strand, antisense strand, non-coding strand, and template strand. Whereas this one, okay, has been called as the plus strand, then coding strand, then sense strand, then non-template strand. So you will ask me a question that since this DNA strand, this one, is being used as a template. Why is it called the non-coding strand? This one should be coding, whereas this one is not participating. This DNA strand is not participating anywhere, but this is called as sense, positive, then coding strand, but non-template. Why first these three nomenclature has been done? Just because of historical reasons and the evidence which are in favor or which is in favor of those historical reasons is the similarity between the sequence of the 5 to 3 prime mRNA and the 5 to 3 prime DNA strand. They are in the same orientation, so it is known as plus strand. They look identical except T is replaced by U, it is known as a sense strand. Okay, and again they look completely identical, they have identicality in their sequence. That is why they are also known as, you can say, coding strand, because the coded mRNA, its sequence matches with this sequence except for T and U, otherwise it matches with the, you can say, non-template DNA strand completely, you can see. But this strand is only a template, its sequence doesn't match with this one. Okay, it is complementary, so it will not match. Okay, it is known as the minus strand, it is known as antisense because it is the anti one, its complementary is anti. Again, non-coding strand it has been termed. So technically it is wrong, but historically this was the reason behind this nomenclature. Now we cannot change the nomenclature. So this is the historical reason, okay, behind this specific nomenclature. Okay, so this is all about the nomenclature of mRNA and the two DNA strands. Okay, so this comes a lot in many examinations, which is the antisense strand, sense strand. From antisense we were told to convert, like in that question you had shown, convert. So those things can be easily solved if you know this concept, if this concept is clear to you. So now we had talked about the promoter sequences. Okay, so the promoter could be, this is your DNA, anywhere could be a structural gene. Okay, this is the first codon starts AUG. Then anything here, minus 10 region, minus 35 region, then minus 55 region. Okay, much upstream is another specific sequence known as upstream promoter element. Promoter element. Now a promoter is said to be a very very strong promoter. Strong promoter means it will cause transcription no matter what. That is the meaning of a strong promoter, means it will cause the binding of RNA polymerase to itself and it will go for, you can say, transcription. Okay. Now such a strong promoter also contains the minus 55 UP element or upstream promoter element along with minus 35 and minus 10 consensus sequences. These are the consensus sequences. Okay, TA, TA, T. Then TTGS here, these are the consensus sequences. Okay, except that there will be again a consensus sequence that is known as upstream promoter because that is present upstream of the promoter, because this is known as the core promoter. These two sequences, they cover up the core promoter. Now this could be the accessory promoter, you can say. So it is even upstream of the core promoter minus 55. So upstream of minus 35 is minus 55. So where the core promoter ends, it is even upstream of that. That is why it is called as upstream promoter element. And if you have upstream promoter element along with both the consensus sequences of the core promoter, the entire promoter region becomes a very very strong promoter. If this minus 55 is missing, like lack of when I'll be teaching you, so in lack of lactosoferon, what happens is the promoter doesn't contain the upstream promoter element. Okay, only minus 35 and minus 10. If only core promoter is present, then it is known as a weak promoter. A weaker promoter is sometimes this sequence is also absent. And this minus 10 is extended. Those are known as minus 10 extended promoters. Those have minus 35 sequence absent. So minus 35 consensus sequence TTGACA is absent. Okay, that is the meaning of, you can say, the -10 extended promoter. These are very, very rare, but these are also present. Okay, so this is your specificity regarding the promoter sequence. Now coming to the hollow enzyme of, you can say, RNA polymerase in Escherichia coli, the gram-negative microorganism that we take as the example, as the exemplar to explain the phenomenon of RNA, mRNA formation, that is transcription, okay, in case of prokaryotes. So the hollow enzyme, which I told you, which I had explained to you in the previous class, the RNA polymerase is only of one type. We had explained it codes for all the three types of RNAs, the tRNAs, the mRNA, and the rRNA. There are multiple other types of RNAs also. You have small nuclear RNA, SNO RNA, small nuclear snRNA, pvRNA. Okay, there are so many types of RNAs are present. MicroRNA, then we have small interfering RNA, siRNA, miRNA. So there are multiple RNAs, but those are not in your course. Only three are there, and only there is one coding RNA. The coding RNA is the mRNA. Always know. Coding RNA is the mRNA. The non-coding RNA means the RNases that, or the RNAs, the RNA molecules that don't code for proteins, that do not code for proteins. Those are known as non-coding RNAs. Like rRNA doesn't code for protein, tRNA doesn't code for protein. Then I told you SNO RNA, snRNA. So those are all non-coding RNAs. Okay, that is again one more style or way of classifying the RNAs. And I have told you already that rRNA is present in the maximum amount, followed by tRNA, then followed by the mRNA in the cell. Okay, in the cytoplasm of the cell. Now, coming to the RNA polymerase, hollow enzyme. Okay, means it will be what? It will be sigma alpha to beta, beta dash omega, because this is the core promoter, sigma factor. Now this has become the RNA Paul hollow enzyme. Now the molecular mass of this hollow enzyme, okay, is 460 kilodalton. Okay, 460 kilodalton. Always remember. This is the molecular mass of this specific RNA polymerase hollow enzyme, means including the sigma factor, is 460 kilodalton. And if I give you a table here, okay, so the genes which code for these subunits are known as RPO genes in E. coli. Okay, just for your information, this is, so like RPOA, A means it will code for the two alpha chains. RPOB, this gene in E. coli will code for the beta chain. Okay, then RPOC will code for the beta dash subunit. Then RPOD will be your sigma subunit. And RPOZ will be your omega subunit. Okay, so these are the genes that code for these specific polypeptides that make up the entire RNA polymerase. Okay, now what happens is the sigma factor is primarily responsible for, I had explained to you that it is responsible for promoter recognition. Okay, even though the catalysis can be done by beta beta dash. Okay, beta beta dash subunit is the catalysis site, means it will add the nucleotides. This one, that is the catalysis, you can say, center of RNA polymerase enzyme. Means this is the active site, you can say, that will lead to the formation of mRNA molecule, means this will cause the phosphodiester linkage. And alpha helps in the binding but cannot cause proper binding without sigma factor. Sigma also, okay, there are many kinds of sigma factors, not one. So, and Mg2 plus is also very critical as a cofactor, okay, for this RNA polymerase to function. Mg2 plus magnesium, it is also a great cofactor here, because an apoenzyme becomes holoenzyme only when vitamins and minerals, okay, coenzyme and cofactor both bind, okay, or one of those bind which are necessary, okay, one of those binds, that is the magnesium, in this case the cofactor, okay. Now when we come to sigma factor, okay, what happens is, after the sigma factor is bound with the core enzyme, which becomes the holoenzyme, the association constant, when we come to chemistry equilibrium chapter, the association constant of the promoter with the holoenzyme, it increases 1000 times, okay, it increases 1000 times. And the half life, means the time taken by half of the molecule to be disintegrated, also increases, means it will stay stably bound, okay, to the promoter. That is also one case, okay. That is why also sigma factor is very essential. So what you have is, for housekeeping genes mainly, okay, there is a concept of housekeeping gene that I'll tell you. Housekeeping genes, the type of sigma required is sigma 70 for housekeeping gene. Now what is a housekeeping gene? So you have prokaryote, you have eukaryotes. So in both these cases, housekeeping genes, it is only a few students know this fact, few people know that housekeeping gene mean different. The term means different when it comes to a prokaryote and when it comes to a eukaryote. In our body, we have so many types of cells, right? A variety of types of cells. Now, I'll give you a simple example, that alcohol dehydrogenase, okay, is produced by liver, which breaks down the alcohol. Will it be produced by the neuron? Brain cells, neuron. Hepatocytes, they produce, but brain cells, neurons, they don't produce it. But the thing is, the DNA is the same, right? It's not that some genes are present in some cells. You have the same DNA, 6 into 10 to the power 9 base pair for diploid, 3 into power 3 into 10 to the power 9, 3 billion base pairs for haploid cells. So what happens is, we have something known as epigenetic silencing that I showed you with stones. So in brain, the stones will be supermethylated, deacylated, so that the RNA polymerase cannot bind to the promoter of the ADH gene in case of brain, and thus the neurons, they do not you can say produce or synthesize the alcohol dehydrogenase protein, whereas in liver it is not methylated heavily, the histone surrounding the promoter of the ADH gene, therefore it is able to cause the synthesis, okay, epigenetic modifications. Now that means what? This gene is not a housekeeping gene, because this is not being uniformly expressed in every tissue, in every cell type. The genes which are expressed uniformly, means every cell type in the body will make that protein from that gene, that is known as a housekeeping gene, like EF1 alpha, which is used in translation, then glucose 6-phosphate dehydrogenase. These are the housekeeping genes, because they are produced in neurons also, heart also, you can say liver also, skin also, muscle also, everywhere. In every cell, this specific protein is produced, means that gene is active, it is not epigenetically silenced in any of the cell type in human beings or in other eukaryotes. So that is the meaning of housekeeping, that it should be expressed by all the types of cells present in an organism. But that is for a multicellular organism. What about the unicellular? Prokaryotes are unicellular, right? If you say all the cells will express that, that definition becomes nullified in case of prokaryotes. It becomes completely nullified when it comes to prokaryotes because they fall under the category of unicellular organisms. Now what is to be done? This question baffles most of the students. Now here the function you might have read in mathematics, so the function of time we will be using. Here we are using function of type of cells, mathematically speaking. Here function of time, okay. So you can understand biology from the mathematical perspective also. You can use function of time. Time means, suppose you have a petri dish, okay, and you are growing bacterial cells, E. coli cells, and they have been provided all the nutrition. Why the nutrient media? This medium contains all the nutrition, okay. Now they are proliferating, fine. But suppose if I take out some nutrients like amino acid leucine, I took out amino acid tryptophan, I took out, okay, like that. Now what they'll have is they'll be under, suppose, some stress, okay, they'll be under stress, and then due to stress conditions or stressful conditions, like if I increase the temperature, increase the humidity, or add acid to the environment, means to the medium, then they'll be having stress. Stress means the, you can say, the outer atmosphere for them, the environment for them is stressful. So now they'll try to survive. Now in order to survive, various other genes, okay, will be transcribed which are not transcribed in normal conditions. So genes essential for survival like hypoxia, you can say related factors. In human it is known as hypoxia inducible factors. So here also different types of genes will be transcribed so as to alleviate the stressful conditions. Okay, stressful conditions. Now, but are those genes always expressed? No, only when stress is induced, then those genes are expressed. So the genes which are expressed all the time, even when you are under stress, even when you are not under stress as a cell, okay. Suppose the, you can say IF1, initiation factor 1 in case of translation, is being expressed all the time because proteins are being synthesized. So it becomes a housekeeping gene for the prokaryotes. In glycolysis, you can say glycolysis always happens, so glycolytic enzymes can become the housekeeping genes. But the genes which are only expressed in times of stress faced by the cells, those genes are not housekeeping genes. Okay, that is how housekeeping gene is defined in case of prokaryotes, and that is how it is different from the definition of housekeeping gene and the concept pertaining to housekeeping gene in case of eukaryotes. Okay, so in case of prokaryotes, only sigma factor is used. So sigma 70 is used for the genes which are expressed all the time 24/7, whether in stress or out of stress. That is known as housekeeping gene in case of prokaryotes. Okay. For other genes, different variant of sigma factor comes and attaches with the RNA polymerase core enzyme. Okay. That was the that is the message that I'm trying to convey. Now tell me, do you feel any kind of doubt or query here? Do you have any query, Ishika, or is it all clear till now, till this part? Any queries, any doubts? Okay, okay, good. So now we'll be concentrating or we'll be shifting our focus to how does, okay, an RNA polymerase find the promoter sequence. Because in eukaryotes, I had explained to you that only 1.8% of the genes are genic, means coding. Okay, genes 98.2 is junk. They're not complete junk, they are non-coding RNAs. but they are out of the reach of your syllabus. Okay, so you can call them non-coding. They will have transposons, retrotransposons, sign line sequences, VNTRs, okay, microRNA, and then mini satellite regions, microsatellite regions. Okay, so those things they'll be having. So that is in case of eukaryotes. In prokaryotes also 40 to 50% is non-coding. Even in prokaryotes, all the gene or the complete chromosome, one chromosome only the E. coli has. Like we have 46, the E. coli contains only one chromosome. Therefore, it is always haploid N, right? So the thing is, throughout the chromosome, genes are not present. Okay. So how does an RNA polymerase find? So one way is it will keep on sliding, okay, from one to the other. One way is it can cause intersegment transfer. Like this is a DNA. This is a DNA. So RNA polymerase is transferring from here to here. It is shifting itself from here to here. That is known as intersegment transfer. Otherwise, it can keep on sliding. Okay. Then next will be it can go for intra-domain association or dissociation or hopping. So it is all very similar to intersegment transfer, but what happens is this doesn't need the two DNA to or the entire DNA strands to fold. Okay, it can occur like this also. Here they are folding. In that case, it can occur like this. Ample distance is there, so it can bind here, then it can just hop. It can then bind here, then it can again hop, it can bind here, trying to find the sequences. So these three are the models, okay, proposed by the scientists pertaining to how does an RNA polymerase find the promoter sequences. Okay, so none has been completely elucidated. Okay, there are still conflicts in research. Okay, so now we'll be coming to the initiation of, you can say, transcription. How does the transcription initiate? Okay, how does it happen? Now coming to the initiation part, okay, what happens is we have something known as the closed complex, then we have open complex, then we have ternary complex, okay, that leads to the, you can say, perpetuation of mRNA synthesis. The first one is the closed complex. Okay. We start with the closed complex. In closed complex, what happens is the DNA remains in the duplex state. Okay, it hasn't been denatured. Means if this remains duplex, okay, then unless this is denatured and it becomes single stranded, then RNA polymerase can read this template strand and make the daughter strand, right? So it needs to be denatured first because this side is the phosphate backbone. So you cannot go for reading from this side. You will see RNA polymerase will read from that side. No, that's the phosphate backbone. Okay. So the thing is, this side is the base. We need to read the base, not the phosphate, because phosphate and glucose or the ribose sugar, the deoxyribose sugar, are the constants. Okay, they are the nucleoside. Okay, so not the nucleoside, sorry. So they are the constant. Nucleoside will be the ribose sugar, deoxyribose sugar plus the, you can say, base, nitrogenous base. But the combination of deoxyribose sugar and phosphate, they are the constants. So you can look here at the structure of DNA. Polynucleotide is DNA, multiple nucleotide. So you can see on in this structure that the ribose sugar and phosphate, they stay at the backbone. Okay, this is the backbone. From here another one will come, hydrogen bonding will occur, and this will stay the backbone of the other, you can say, strand as well, right? So the thing is, you can look here also, you can see backbone, backbone. So you cannot read it from this side. You have to denature this hydrogen bond and then read from here. You want to read the bases because that is constant. What you will do? What you will do? The DNA sequence is the base sequence because deoxyribose sugar and phosphate group are constant. They are not changing. It's not ATGC there. Okay, everywhere they are the same. So it's the AT adenine, guanine, cytosine, thymine that will change the course of the, you can say, or that will determine the course of what amino acids will be coding because of the 3 codon rule, right? Now, closed complex means the denaturation has not occurred, means RNA polymerase has not used its helicase activity. So, DNA polymerase, okay, is assisted by, when we look at DNA replication, assisted by DNA helicase. Okay. That is known as DNA-C in case of prokaryotes and MCM complex. Okay, MCM 2 to 7 in case of eukaryotes. Mini chromosome maintenance complex or DNA-C. It uses ATP. In case of DNA polymerase, the helicase enzyme is a separate enzyme. It uses, DNA-C uses ATP and uses the energy produced after dissociation of ATP into ADP plus PI to go for, you can say, energy formation, and that energy is used to break the hydrogen bonds. Okay. So in case of RNA, the RNA polymerase does possess helicase activity on of itself, okay, and it's known as the closed complex. In the beginning, it will just bind to the DNA, okay. The complex will be reversible, okay, in the beginning because the equilibrium constant will not be very high, okay. Now the closed complex is converted, okay, into an open complex by, you can say, it is an open complex of 1.3 tons of double helix in a series of steps by melting. Melting means denaturing the short DNA region, okay, around the minus 10 region, okay, around the minus 10 promoter region, okay. First it will lead to an intermediate formation which is little bit unstable, but once it has been formed, the closed to open complex transition is irreversible. It gets stabilized as the forward rate constant is very high. Okay, Kf, you can say, as per equilibrium, is the forward rate constant is very high for the open complex. Open means now helicase activity would start. Okay, so Kf would be high. Helicase activity will start or initiate. Okay. And then what will happen is it will start from denaturing the minus 10 promoter region, that is TATA AT region. Okay, so the thing is, if you analyze it, you will see that changes in the shape of RNA polymerase will change its shape, okay, when it's going through the transition. It will cause conformational change in its shape, and that will lead to change in the kinetics. Okay, kinetics means Kf increases the kinetics of the reaction, the rate of the reaction. So in a closed complex, I'll tell you, if you compare closed versus open, okay, in a closed complex, the RNA polymerase binds from minus 50 to plus 1, that is extends to about or covers about 55 base pairs. So it binds to, this is the plus 1 first one, here is the minus 55, means still the UP element. So RNA polymerase will cover like this. Okay. So minus 55 to plus 1, almost 55 bases it covers. That is the role of minus 55 region. Now, the double stranded DNA binds primarily along you can say one face of the holoenzyme to one side, okay, which contains the, you can say, C terminal domain, which means the domain present at the C terminal. And domain is an entity of the enzyme which has its unique property. Okay, so domain. So there are two things in an enzyme. You have motif, you have domain also. So many times in the books it will be asked, like it will be present that it has an N terminal domain, it has a C terminal domain. Okay, so they'll be asking questions regarding a domain and a motif. So a motif is what? A motif and domain both are parts of the enzymes or proteins. So a motif is a short conserved sequence associated with distinct functions. Okay, it will have a distinct function. Okay, but if you isolate this specific, suppose from here to here you have a motif, if you isolate this from the protein, it won't be able to function independently. So function independently cannot happen. Okay, when it comes to a motif. Now, when you come to a domain, okay, so a domain is a bigger unit of a protein with more polypeptide, you can say more amino acids pertaining to it. So it is also a conserved sequence pattern. Okay, similarities both are conserved. So I had explained to you the meaning of conserved in the last class. They have they are both conserved sequence patterns. Okay, pattern I'll be saying pattern, but it is defined as an independent functional and structural unit. Means it can function independently if it has been isolated from the polypeptide chain. If you cleave and isolate it, if you break the peptide bond and isolate the domain, so it would have its proper stable quaternary structure by which it can still perform its own function. So independent functional and structural unit. Okay, the meaning of that is, and also it is longer than the motif, just like I told you. So I'll write it down also, it's longer than any motif. So mainly what you will be reading about is the C terminal domain towards the C terminus, the NTD or the N terminal domain pertaining to RNA polymerases. So it binds to DNA via its C terminal domain. Okay. Now, after the transition happens from the closed to the open complex, okay, what happens is the conformation of both the RNA polymerase and the DNA, it will change. Okay, the conformation of both the RNA polymerase and DNA change. Now, the most dramatic changes, okay, in the structure of the RNA polymerase DNA complex, when we talk about the open complex, the most dramatic changes occurring in the RNA polymerase promoter complex are, okay, change number one, what will happen is an approximately 90-degree bend in the DNA occurs. Okay, and why does this bend occurs, or why does this bend occur? An approximately 90-degree bend in the DNA occurs. Again, the reason is, the question is why. Why does this bend occur is when you bend the DNA 90 degree, it will allow the template strand to approach the active site of the enzyme RNA polymerase. Okay, it will allow the template DNA strand, or simply the template strand, okay, to approach or be proximal to. Proximal means very very close. Distal means far. Okay, be proximal to approach the active site, because we all know that the active site of the enzyme catalyzes the reaction. So it must approach the active site of the enzyme. Enzyme is RNA polymerase itself. So that is why this is the first change that occurs. DNA will be bent by 90 degree, or at an angle of 90 degree. Now, strand opening means denaturation is happening of the promoter DNA starts from -11 to +3, means it will involve the -10 region, just like I had told you. So strand opening means denaturation, helicase activity uh, is being happening here, or has been made to be implemented. Okay, so strand opening of the promoter DNA between, you can say -11 to +3. +3 means where the real structural gene is. +3 with respect to the transcription start site, means from +1 transcription starts respect to the transcription start site TSS. Okay. Point number 3 is what will happen is scrunching of the promoter DNA into the active channel, means it will be scrunched, means it will be bent and put the promoter DNA after 90 degree angle bend also. Okay, it will come close. After that, the promoter will be further scrunched. The promoter of the DNA will be further scrunched, further you can say squeezed, and it will be put into the active channel so that the active channel can grab the entire promoter. Okay, so scrunching of the promoter DNA so that the binding is proper and the association or recognition is proper of the promoter DNA sequence, okay, into the active channel that forms something known as transcription bubble, or into the active site, into the active site which forms the transcription bubble. Very very important is transcription bubble. So what happens is, suppose this is the active site, okay, of RNA polymerase. So now this was the promoter sequence. From here we have -35, we have -10. So what will happen is this will be scrunched like this to be able to fit in -35 like this. So you can fit in -35, -10 here only, so that it can read it properly, and then binding will happen properly, and then catalyzation, or sorry, catalyzing of the site or of the reaction will happen at an appropriate rate. So now DNA will be state, it will be scrunched and squeezed into the active channel or active site of the RNA polymerase. Now after that, the enzyme will also undergo conformational change. Suppose this is the enzyme. Okay. Now the enzyme will close its jaws like this around it. This is the active site, so the enzyme will close its jaws around it, and you have the promoter DNA inside. So I change the color. Now this is the promoter DNA inside. The enzyme will close the jaw. Okay, this is the DNA. This is the RNA polymerase. Okay. So the fourth point is, you can say, the closing of the jaws of the enzyme. Closing of the jaws of the enzyme means RNA polymerase of RNA polymerase to encircle this section of the promoter. If at all any promoter exceeds downstream of the transcription site. So there are those kinds of promoters also, very special promoters. We can write down and circle the promoter which are downstream of the transcription start site. So that is how we go for it, and therefore the promoter will be scrunched. When they are scrunched, what will happen? More sequences can be bound, right? You just look at this. If they were like this, it would have bound from, suppose like this is the promoter. Now at this stage from here to here it is binding. Now it is scrunched like here. So completely here to here it is bound. So more base pairs are covered. So in the open complex, the RNA polymerase binds to, you can say, 75 base pair, not 55. 55 was in the open, sorry, closed one. Here it will bind to minus 55 to, you can say, plus 20. Okay, total is how many? 75 base pairs. So 20 compared to 55 base pair bound at the or during the closed complex, 75 base pairs are being bound in the open complex. So an increase of 20 base pairs. Okay, that is why scrunching is done, so that more of the bases can be bound, okay, after the denaturation. That is the open complex. Okay, and after this open complex is done, or after this open complex is formed, the start of the ternary complex will occur. Okay, now the next step will be to incorporate the first two nucleotides and form the phosphodiester bond between them, and this will generate the ternary complex containing RNA as well as DNA and the enzyme. Ternary means 3. Now after this you will be having the ternary complex, means ribonucleotides. Okay, NTPs will be produced, will be arriving as the substrate. So two NTPs will be arriving, and then from 5 to 3 prime end, phosphodiester bond will be produced, will be formed. So phosphodiester bond is formed, and then what you have is ternary complex. Why? Because ternary complex means 3. You will have now RNA in the complex, you have already DNA in the complex, and you have RNA polymerase in the complex. Now these 3 molecules, okay, or this RNA, DNA and the enzyme will form the ternary complex. Okay, now one thing I'll tell you here, if you compare it with DNA replication, you don't need any primer here, means you don't need a pre-made stretch of 5 to 3 prime nucleotides, suppose 8 nucleotide sequence or 5 to 8 nucleotide sequence, which will provide a free 3 prime OH. Okay, because this is needed by DNA polymerase. DNA polymerase cannot synthesize anything de novo like RNA polymerase just did, means on its own, out of the scratch. It needs a 3 prime OH group, free 3 prime OH group, and then when deoxyribonucleotides DNTPs come in, then it can catalyze its addition. But if you just keep two fresh DNTPs, it cannot start. It cannot initiate the formation like RNA polymerase did. It cannot do it. Okay, so RNA polymerases, one mark question, they do possess, even eukaryotic RNA polymerases, they do possess the de novo polymerase activity, but the DNA polymerases, they lack the de novo polymerase activity. Thereby, they require a primer, a short stretch of nucleotides, 5 to 8 nucleotide sequences, to provide them free 3' OH group. To that free 3' OH group or end, okay, to this end, now the deoxyribonucleotides can be attached. Okay, so that is the main role. So now tell me, Shika, do you have any queries in this lecture? Any queries, any clarifications required till the ternary complex formation? Now we have started to produce mRNA. Okay, so this is how mRNA is produced in prokaryotes, and similar mechanism is also uh, similar mechanism occurs in eukaryotes as well in our body too. Okay, then good. So stay safe. Have a nice day, and if you have any worksheets, again you can send it to me on my personal number. Okay then. Thanks a lot. Let's meet you in the next class.